Okay, this is the last section of the introduction to vision and really ending up with the properties of the photoreceptors, which we had done quite a bit of. We had gone through the rods and the cones and some of the differences between the rods and the cones and how the characteristics of the rods versus the cones and their distribution and so forth really shape our experience of the visual world. And just so, again, to put us back where we were, I know there are a couple pictures that I keep putting up. Usually it's where I ask students to think about questions, but that we have the photoreceptors are doing that job of transduction, and that for the most part, there is quite a bit of convergence between the photoreceptors and the ganglion cells, as we have 126 million of those photoreceptors. We need to get that information out of the eye through the axons of the retinal ganglion cells, and there's only 1 million of those. So we're going to have to see this kind of convergence and spatial summation, particularly for the rods, more so than for the cones. And of course, the foveal cones have that one-to-one wiring. And we will get to those horizontal cells and amacrine cells doing the crosstalk there as we move forward. So what the photoreceptors are doing at the very back part of that retina is they're doing the job of transduction, right? Changing that light energy into electrochemical energy for the brain. We're going to go through the structure of a rod briefly so that we can talk about the structure that exists for transduction to occur in the photoreceptors. And this is showing the basic structure of a rod photoreceptor, but the cone is really quite similar, where it starts where we can talk about the inner segment, so where we really have the cell machinery, the outer segment. So if you look over to the right, but you're in A, in the first part of the picture, the outer segment is that top part that is showing these discs, these stacked discs. And at the very bottom of that picture is the synaptic pedicle, where it's going to communicate on to other cells, to the bipolar cells, or to the horizontal cells. Those outer segments contain these stacked discs. So each disc is going to contain thousands of visual pigment molecules. And these are the visual pigment molecules are actually the molecules that are doing the transduction. So what's being shown in B is this magnification of parts of the two of those discs coming together, and you can see that where the membranes of those discs are meeting, the visual pigment molecule in the picture, it looks kind of like a tack. It crosses across that membrane, and that visual pigment molecule, so if you look down at the bottom and see, it is this long protein strand of opsin, and that opsin is going to snake across that membrane 7 times. You can kind of count that there if it's going across that membrane 7 times. And then the opsin is connected to 1 molecule of retinol. So each visual pigment molecule is made up of 1 unit of opsin and 1 unit of retinol. For the rods, that unity, those two things together are called rhodopsin, which really the rhod, R-H-O-D, is for red, and we'll see in a minute that it has this reddish appearance. That retinol, that is actually the molecule that is doing the absorption of the photons of light. So in the last picture, it showed the opsin snaking through that membrane 7 times. You could see that it was really quite long, and that the molecule of retinol, that little blue dot on there, that it was really tiny in comparison to the opsin. And now what this picture is showing is really a part of the opsin magnified. So this is one little splice of opsin, so that now we can really see the retinol. Okay, and you can see that when the visual pigment molecule, when the eye is in the dark, that retinol is bent over, kind of clinging on to the opsin. And when we put that molecule, or in the eye, in light, the retinol isomerizes. Okay, it isomerizes meaning it changes shape, it straightens up, and it breaks off from the opsin. So if we look at the process of transduction, the light is absorbed by the retinol undergoes isomerization, it changes shape, the bond between the opsin and the retinol is broken, that retinol breaks off, and then it affects other enzymes, or these messengers that cascades that then influence other visual pigment molecules to break off this way and isomerize, and which also then influences other enzymes. So it ends out affecting the entire receptor. So one photoreceptor can have millions, basically, of these visual pigment molecules. One photon of light is absorbed by one retinal molecule, and that can cause the whole photoreceptor to fire through this because of this enzyme cascade. And I'm talking about this, and I talked about this with the wiring as if the photoreceptor is firing. That is how your book talks about it, so I want to leave it as simple as possible, but I also don't want you to go on to another class and think, wait a minute, that's not what they were saying. Actually, things behave very differently in these photoreceptors, and they are inhibitors. They are when they're firing, they're inhibiting the bipolar cells, and so what's happening with all of this is actually a turning off momentarily of the photoreceptor, so it stops that inhibition. But for the most part, for this class, I'm going to ignore that because it makes it harder to talk about all of these processes for me. So we see this kind of visual pigment bleaching, and then those visual pigments have to regenerate in order for them to be used again. So we get one photon of light, the retinal absorbs the energy, the retinal isomerizes, it changes shape, it breaks from the opsin, that triggers that electrical event, that cascade of enzymes that are affecting other visual pigment molecules, and this is causing transduction, basically. And if you look down at the picture to the right, and it goes A, B, C, A, and at the top of that, it's showing how the opsin and how the retinal is bent over and attached to the opsin. So that is the molecule in the dark, and you can see that kind of reddish appearance. So the bottom pictures of that are of a frog's retina. And then as we put that those molecules in light, you can see in B the retinal straightening up, and then in C it's broken off. And as we're getting to see where the eyeball is in light, that we have this kind of bleached appearance, which is why that's called visual pigment bleaching. So right now, with the retinal broken off and this isomerization process having occurred, those photoreceptors cannot be used again until they regenerate and they go back to the position in A where the retinal is attached and bent over. So we have to go through this visual pigment regeneration. And this takes enzymes. It takes enzymes from the pigment epithelium that act as a catalyst for the retinal and the opsin to now reunite so that we can go through that again. It takes time, this regeneration process. For the cones, it takes about 6 minutes. For the rods, it's estimated somewhere usually between 20 to 30 minutes. That very last piece of our photoreceptors in the review, now that we've really gone through transduction and how they work, is to come back to this idea that they differ. Rods and cones differ in their functional significance. So rods are really helping us to a large extent much more for our dark adaptation. And if you see that visual pigment bleaching and that it takes time to regenerate, it takes time to see in the dark after being in the light. And the way they do this, they've done this research. I'm going to start with this picture on the left of, they're going to have somebody looking at a fixation point, and then they're going to have a test light that's off in the periphery. So they need to keep their eye, their foveal vision on that fixation point so that that test light is off in the periphery. That's how we're going to start to look at dark adaptation and these dark adaptation curves. So this picture shows several different dark adaptation curves, and I usually do use a pointer quite a bit here, so I'm hoping that this works out okay for me just talking over a slide. But you're going to be looking at first the red line. It kind of turns purple at the bottom, but the red line that has the kind of almost weird W shape. So what I'm doing, remember, I'm having the person look straight forward, and there's a flashing test light in the periphery. And so remember that what this means is they are perceiving that flashing test light with both rods and cones, because both rods and cones exist in the periphery. Okay, and they're going to start off in the light. So if you actually look at the dots there on the far left on the y-axis, the one that's lower says the cone light adapted sensitivity. So when we are in the light, we're really using our cone vision. And when we test those people with how low can they make that test light and still see it, so we're getting their threshold, right? Their absolute threshold. This is the example of when they've used that method of adjustment. They're just making that test light so they can just see it. Okay, they're pretty high on that figure, meaning they have a high threshold, low sensitivity. Okay, so we turn off the light and they can make that flashing test light slightly lower, and that's what's happening with this red curve, and we're going to watch that over time. And you can see they can make it slightly lower and slightly lower and slightly lower, again with the red curve, and they do this until about 6 or 7 minutes out, and it starts to level off. And if you remember, the time for cones to regenerate was about 6 minutes. So our cones have regenerated, and we see this kind of leveling off, that this is really as good as we're going to get with our cones. And then at about 12 or 8 or 9 minutes, maybe 10 minutes, we start to see they can make that light lower again, and lower and lower and lower, and they can do this for where they're getting more and more sensitive. They get a lower and lower threshold up to about 20-25 minutes out. And when they have, this is as low as that's going to go, and we see another leveling out. Okay, so this is with people with, quote, normal vision, looking at this in the peripheral vision. So using both their rods and their cones, we see this kind of dip, a leveling out, and then this dip and a leveling out a second time, where they get pretty high sensitivity. All right, I'm next going to show that this is because of the cones and the rods regenerating, and so giving us more and more ability to see. And I'm going to start, I guess, with the cones. Okay, so if you look at the green line, this is using the fixation cross. You're now looking at it with your foveal vision, and the flashing test light is occurring right there in your foveal vision. So this is just using your cones. And you can see they have that same starting point in the light. We turn off the lights, and they have that same starting point where they have pretty high threshold. They're slowly lowering and lowering that test light. It levels off at about 6 or 7 minutes, and it stays there. And that's really our maximum cone sensitivity. They've also looked at the pure rod curve. So with this, they had to get people who are monochromats, who have no cones in their eyes at all, and they only have their vision is based on is a rod-based vision. And you can see, first of all, with the dots to the left on the y-axis, that rod light adapted sensitivity. So this is again off in the periphery, but they have no cones, so this is just their rods. You can see that their threshold is much higher than the cone light adapted sensitivity. They are really not very sensitive to this test light. We turn off the lights. You can see it takes a little time before they even start lowering test light at all. And then that lowering, it goes down and down and down in this really, and that kind of blue curve is really, there aren't any kinks, or I'm not doing a very good job with my words, a really flowing kind of curve, right? And at some point they reach the people who had their maximum cone sensitivity, and then they get better and better, and they match with the people who were using their rods, who were able to use their rods when we were doing that in the periphery, and we were using both rods and cones. So you can see some of that, what's going on with the people who are monochromats and just using their rods. Some of that is going on in the background for all of us, but since we have our cones, they're giving us more sensitivity. Okay, I'm hoping that came out okay with these dark adaptation curves. It's always kind of a complicated thing for me to talk about, even with using my pointers and everything, but do think about asking questions when we come back on Monday. As we get to the functional significance, so for rods, they're really giving us that good dark adaptation and the ability to see in the dark. For cones, they are really giving us our color vision, and we're going to see how they do this by looking at spectral sensitivity curves. So the curve that shows the relative sensitivity, but usually what it's showing is the absorption spectra for a particular type of cone or for the rods. And this is just showing the rods are better at about 510 nanometers. That's where they're doing their maximal absorption, or where they have their most sensitivity. And the cones have their maximal absorption, their best sensitivity, right around 560 nanometers. And how do we measure spectral sensitivity? Well, we use now what's called a monochromatic light, which monochromats, the people, it means they have one chroma, one ability for a particular kind of wavelength, so they're only really using their rods, usually is how they work. But a monochromatic light means one monochroma, one color light. So this is truly one of those sinusoidal waveforms that is showing us one single wavelength of light, so that we can determine a person's threshold at different specific wavelengths without those kind of mixes that are usually going into the colors that are our shirts and books, and the colors that are made from things in the world. The threshold for light is lowest in the middle of the spectrum. You can see right around 550 nanometers. We have our lowest threshold, which is up in the top of this figure. And if we flip that around, so the 1 divided by threshold is our sensitivity, we have our greatest sensitivity then as well, right around 550 nanometers of light. And how the cones give us this color vision is because we have 3 different types of cones. We have short wavelength cones that are absorbing maximally at about 419 nanometers, medium wavelength cones marked there with the M, absorbing maximally around 531 nanometers, and then long wavelength cones absorbing maximally around 558 nanometers. And then we have our rods in there at about 510 nanometers. So we're going to talk about this much more in color vision, how we use those ratios of the different absorption levels, the different responses of those cones in order to give us the experience of color. In this review of the photoreceptors, of course, I've put this up here a few times now, the differences between the rods and the cones are kind of summary. We have differences in physical appearance. They're different in how they are distributed across the retina. They are different in their wiring to the brain, which explains the differences in acuity and differences in sensitivity to dim light between the cones and the rods. They are different in their clinical issues, which we saw, which gave us some hints at the differences in their functional significance, that they have these different functions, because the people who were losing their cones from macular degeneration, they were losing that acuity, they were losing their color vision. People who were losing their rods from that genetic disorder, retinitis pigmentosa, they were losing their rod vision. They were really having more difficult seeing at night, seeing dim lights.